Welcome to another exciting episode of Inside Learning. Today we are going to discuss about F positive, F negative, HFR and F prime factor in the process of bacterial conjugation. Conjugation is the method of exchanging of genetic material between the two different bacteria of same generations. We are talking about the siblings. We are talking about the bacteria that are different but they belong to same generation. Both of the bacteria that are going under the process of conjugation belong to the same generation. The process of conjugation is also known as horizontal gene transfer. It is also known as horizontal gene transfer. Horizontal gene transfer means the process that took place in the exchange of genetic material is in horizontal plane. The process of conjugation involves the direct contact between the two bacteria. Direct contact or the direct physical contacts between the bacteria that are undergoing the exchange of genetic material. Conjugation is took place by mean of plasmid. What is plasmid? Suppose this is a structure of bacteria. In bacteria we have the chromosomal part. This is the chromosome of bacteria, but here is also another segment which we usually call as extra chromosomal material. We call it as plasmid. The extra chromosomal material present in the bacterial cell is known as plasmid. This is plasmid, the extra chromosomal material. So the process of conjugation is took place with the help of plasmid. Why the process of conjugation is even taking place? Reason इसकी क्या Reason why the conjugation took place is because antibiotic resistance. See, some of the bacteria are antibiotic resistant. They show the resistance towards the antibiotics. So every bacteria should attain this quality to maintain its life. So in order to do so, the genetic material, the extra genetic material plasmid, which is responsible for the antibiotic resistant in a bacteria, is transferred to other bacteria so that it may also be resistant to antibiotics. Antibiotic resistance. That's the reason the process of conjugation took place. So here are some of the basic points. What is conjugation? Conjugation is the exchange of material between the bacteria of the same generation. There is no parental or offspring generation. I am talking about the sibling. They are of the same generation. They are of same age. The bacteria belongs to the same generation. Second, they have the direct physical contact in order to exchange their genetic material. Conjugation is something that usually involves the direct contact between the two bacteria that is undergoing the process. It involves the transfer of plasmid. The whole process of conjugation is took place with the help of plasmids. It occurs because the some of the bacteria are antibiotic resistant. So in order to convert all the other bacteria in its antibiotic resistant state, it needs to be transfer plasmid from one bacteria to another bacteria. This is the reason why whole the process took place. Let's just talk about the process of conjugation. Consider this as a bacterial cell. This is bacterial chromosome, normal bacterial chromosome, and this is plasmid. In some bacteria, there are F plasmid. F is the term that is the abbreviation of fertility. We can call this as fertility plasmid. F stands for fertility. Some of the bacteria contains F plasmid. Other bacteria contains only simple type of plasmid. The F plasmid stands for fertility plasmids. Fertility means this bacteria is able to make a specialized structure named as sex pili. This is basically the structure that it is involved in the process of conjugation. So some of the bacteria who are able to perform the process of conjugation have this F plasmid in their structure or in their cell. This is called as the sex pili, or we can also call it as conjugation tube. The bacteria contains F plasmid have the ability to form this sex pili, which is involved in the process of conjugation. We can also call this cell is also known as F plus cell. because it has that F plasmid, the fertility factor or the fertility plasmid. It is also known as F cell. Now, considering it the main cell that is going to transfer its F plasmid, basically the plasmid F plasmid is the one that is going to transfer from one cell, bacterial cell, to another bacterial cell. Now suppose if we have an other bacterial cell from the same generation here, it is the normal genetic material of the bacterial cell. There is no F plasmid. It may have some other kind of plasmid, some normal plasmid, but not the F plasmid that is involved in sex life formation. This cell, because it lacks the F plasmid, it is known as F negative cell. And as we know that the F plasmid or the extra genetic material, antibiotic resistant chromosome, is going to transfer from this bacteria to this bacteria. So this cell, this bacteria or this bacterial cell, the F positive cell, is also known as the donor cell. Donor. It is going to receive that, so it is also known as recipient. It is known as recipient cell. So they call F negative cell because it don't have fertility plasmid in it. Once the F positive cell form its sex villi, it start to generate a connection between these two cells. So in the next step, they are come in close with each other and start to transfer the genetic material. This is the F positive cell, this is the F negative cell, and they connected by with the help of conjugation tube. Now this is known as conjugation tube. The tube that is involved in the process of conjugation, that is involved in the process of exchange of genetic material. This is the chromosomal part, this is the plasmid, and it's going to be transferred here. It replicates. Plasmid is a self-replicating material. This is the new structure in this cell, which is called as F positive, yeah, F plasmid, and this is the chromosome of this bacteria. So they now seems identical. The plasmid, the F plasmid, is now transferred to the F negative cell, and ultimately this F negative cell will convert into F positive cell because now it has that F plasmid. Whenever we call some bacterial cell F positive or F negative, it is based on the presence of that F plasmid. If F plasmid present, we call it as F positive cell. If not present, we call it as F negative cell. Now the F negative cell is converted into F positive cell because it has that F plasmid now in its structure, now in its wall, under the wall. So we can say that the first type, here we can write the type of conjugation. Type. So the first type and result, I must say. So the first type is F positive. Parent was F positive and F negative, and as a resultant we have F positive. Both the cells are converted into F positive cell. So this is the first type of conjugation. Now we discuss then other type. There may be some other condition in the process of conjugation or during the process of conjugation. Suppose the genetic material is transferred, the plasmid is transferring from F positive cell to F negative cell, and the process took place some time. So meanwhile, all the surroundings around these two bacteria, there are also some external factors that are going to affect the process of conjugation. So if during this whole process, this is the chromosomal part of this bacteria, this is the chromosomal part of this bacteria, this is plasmid, new plasmid that is going that is transferring in this cell, and this is the plasmid of F positive cell. So during this whole process, some external factors also affecting the process of conjugation. If somehow the conjugation tube breaks during this process due to some external factors, and the plasmid that is transferred is not transferred completely, and it is half, like it's supposed to transfer the whole plasmid, but duplicated whole plasmid, but it transferred only half, and suddenly the conjugation tubes breaks. So then what next happens? So there is two possibilities after that, two possibilities with this plasmid segment. First possibility is maybe destroyed, this plasmid part that is transferred in F negative cell is completely destroyed, or further it has no life. Destroyed, it may be destroyed completely, or the second option, it may be enters, or it may be integrate, or it may become the part of chromosomal segments of this DNA. The bacteria that has this segment of plasmid, new segment of plasmid, it may destroy this with the help of nucleases enzymes. It transferred some destroyed by some external factor, only segment of plasmid transfer. It may be destroyed by nucleases, or it may try to adjust in the bacterial chromosome with the process of homologous recombination. So it may be destroyed, it may be integrated into this chromosome. So if it is integrated, what would be the condition look like? This cell will now look something like this. This is the chromosome, bacterial chromosome, and this dotted, I'm representing it with dots. These dotted segments show the plasmids, the plasmid that enters that integrated into the chromosome of bacteria. So now the bacterial chromosome is completely changed. It is an other whole different condition. It is not only that plasmid is present along with the whole chromosome in the bacterial cell. It is that chromosomal bacterial chromosome is converted, is recombinant, is changed into something different. This condition or this type of bacterial cell is now known as HFR. Is now known as HFR. What is HFR? HFR means high frequency recombination. It has higher tendency to transfer genetic material to fellow bacteria. Why the higher frequency recombination means? HFR bacteria, what is the property of HFR bacteria? HFR bacteria are usually those bacteria that are capable to transfer the genetic material at greater frequency. See, there are some bacteria which are F positive cell, but there are some bacteria who are HFR. HFR are greater in tendency, or they have the ability much larger than F positive cell to transfer the genetic material from one bacteria to another bacteria. So this is the HFR condition or HFR bacteria. The second condition, AF positive and F negative type forms the result of F positive HFR. Maybe it is F positive if it continuously in the normal process, and if not, it may synthesize the HFR. Now the last type we are going to discuss of conjugation is, now the HFR bacteria is always trying to get rid of that plasmid chromosome. It always try to get rid of that accumulated or integrated part. So in order to get rid of that plasmid, F1 HFR resulted converted into F prime factor. How? Here I am showing you. This is the bacteria, this is the HFR bacteria. HFR. This is the chromosome of original bacterial chromosome, and the dotted one segment I am showing here, this is the integrated plasmid. So the integrated bacterial chromosome is always trying to push or pinch out, like this, pinch out the segment of plasmid chromosome in this way. I am showing you here. This is the bacterial chromosome that pinch out the segment of plasmid chromosome, like in this way. So ultimately, when the segment of plasmid chromosome separate from the chromosome of bacteria, it takes some part of the bacterial chromosome along with it, like the one part, like a little segment of original bacterial chromosome is also pinched out with the plasmid chromosome. This little segment of bacterial chromosome, not only the plasmid chromosome separated, but also a little part got separated from its original bacterial chromosome. So as a result, now it has, the bacterial cell has, this is whole bacterial chromosome, and this is the plasmid chromosome, the segment of plasmid which is separated along with the little part of bacterial original chromosome, this way. So now this is the whole new structure, this is the whole new possibility of bacterial structure of bacterial chromosome. Now this bacteria is known as F prime factor. This bacteria is known as M prime factor. M prime factor has original bacterial chromosome plus plasmid segment along with the bacterial chromosome integrated into it. This is a whole new condition. So we can say that when the third type of conjugation is when HFR is trying to convert into a F positive, it may sometimes due to some errors converted into F prime factor. Suppose if it doesn't happen, the plasmid that is pinched out from the original bacterial chromosome does not contain any of the part, or does not come out along with any of the part of chromosomal bacteria, it will definitely convert into F positive. So whenever HFR is trying to convert into F positive bacteria, F positive cell, it sometimes due to some conditions, due to pinch out or due to some external factors, are converted into F prime factor. I think now you have the clear concept of what is F positive cell, F negative cell, what is HFR cell, and what is F prime factor. HFR cell is something when bacterial chromosome has integrated plasmid chromosome. And why this happened, I already have explained in the process. So thank you for watching my video. If you like my video, do subscribe to my channel.